Conversations regarding dystrophinopathy often include discussion of what a person's genetic variant is and how that may or may not impact their treatment options. The goal of this video is to provide an overview of the different types of variants in the DMD gene and what impact they can have on dystrophin protein production, along with the variety of treatment pathways for dystrophinopathy. You may have heard your DMD variant referred to in many ways: a variant, a mutation, a change in the gene. These terms are all referring to a difference in the DMD gene from what is expected, which may impact how the gene is functioning. There are thousands of different variants that have been reported within the DMD gene, but we can group them by type. The most common types of variants involve a deletion or duplication of one or more exons of the DMD gene. Smaller variants can also happen within an exon. This includes nonsense variants, which cause a premature stop in the gene, or a missense variant that changes the amino acid that is added onto the growing protein. Some people have deletions or duplications of one or more bases, the small letters that make up the DNA within an exon. There are even variants that happen outside of the exons, in the non-coding areas of the gene called introns, which can change how the exons are coming together. Some people have other, more rare variants as well. If you are unsure what type of variant you or your loved one has, reach out to your care team to discuss or take a look at your genetic testing report. Here we can see some examples of what different types of variants might do within the gene. Let's say the sentence, "The old dog ran too far," is part of the DMD gene. You can see that a deletion would take out part of the gene, a duplication would duplicate part, a nonsense would end the sentence early, a missense would alter one of the words, and an insertion would add something new. Just as changing the words or letters in our example sentence can impact its meaning and whether it makes sense, changes within the DMD gene can change how it is functioning, and certain changes can have more impact than others. Following a positive test result, you may wonder if a genetic variant can determine a person's diagnosis and progression. This is known as genotype-phenotype correlation, where someone's genetics, or genotype, may determine their phenotype, which includes their symptoms and how they change over time. And the answer to this question is sort of. Providers and researchers can look to the type of variant a person has and try to interpret how that variant may affect the gene's function. In DMD's case, to what degree that variant is expected to change our production of dystrophin. However, it is important to note that there are always exceptions to the rules, and each person may present and progress in a unique way. The type of variant a person has, or which areas of the gene are involved, can shine some light on the expected impact of the variant on symptoms. For example, does the variant cause the protein to be stopped early, or does it allow for some dystrophin to be made, but maybe it is shorter or structured differently? The variant outcomes can impact dystrophin, which can in turn affect a person's symptoms. Talk with your healthcare provider about your genetic variant and what has been reported before to see if they are able to provide additional insight. Variants that are in frame may have a different expected effect on dystrophin production compared to variants that are out of frame. But what do these terms mean? The reading frame is referring to the structure, starting points, and stopping points which allow for the body to pull instructions from the DNA to make a protein. Changes to the reading frame of DMD can happen from deletions or duplications of entire exons or smaller variants within an exon. Let's look at a few examples. You may remember that the DMD gene is made up of 79 exons that fit together like puzzle pieces. When we talk about deletions or duplications of one or more exons, you will often hear about the reading frame rule. When our body is making dystrophin, the exons of the DMD gene fit together and the instructions are read for producing dystrophin. When there is an out-of-frame deletion or duplication of one or more exons, the two ends bordering that section can't fit together. This results in little to no dystrophin production and is more commonly associated with a diagnosis of Duchenne. You can see this illustrated by the example sentence here. If you delete dog from the old dog ran, old and ran can't fit together, so our protein stops early at the old. If the deletion or duplication of one or more exons is in frame, the exons bordering that section can fit together, and some dystrophin can be produced. This dystrophin may be shorter and have varying levels of function. In-frame deletions of one or more exons are more commonly associated with a diagnosis of Becker. You can see this time the deletion includes old, but the and dog are able to fit together, making a shortened version of the intended sentence. Frameshift variants can also happen within an exon. You may remember our genes are made up of 4 DNA bases, which are read in groups of 3 to make an amino acid. Additions or deletions of those letters within an exon may cause a frameshift that leads to a very different structure to the protein being made, or an early stop in the protein production. You can see that sometimes deletions or duplications, whether they contain a whole exon or happen within an exon, can cause an early stop of the protein. That is why a nonsense variant can have a similar effect on protein production to a frameshift variant, because it also causes an early stop of the protein. Now that we have covered genetic variants, let's look at treatment options and how some of these may connect back to genetics. There are many different ways that treatments can target the symptoms of dystrophinopathy. However, they generally fall within 2 larger categories. Some therapies attempt to restore or replace the absent or altered dystrophin. Other therapies try to prevent or alleviate the symptoms in the body due to absent or altered dystrophin. It is important to note the category of restoring or replacing dystrophin is the only therapy strategy with variant exclusions or limitations based on genetic variant. All other therapeutic approaches are not limited by genetic variants. Looking closer at restoring or replacing dystrophin, we can divide this into 2 strategies. Replacement therapies, such as gene therapies, are not variant specific, but may have variant exclusions or specific variants that are not eligible. This may include higher safety concerns for individuals who have variants within a certain region of the gene. Restoration therapies include things like exon-skipping and read-through therapies, which are targeted to a category or specific set of genetic variants. Exon-skipping therapies, of which there are multiple FDA-approved here in the US, utilize the reading frame rule we discussed earlier to target out-of-frame deletions of one or more exons. These therapies skip over an extra exon that borders an out-of-frame deletion. Though this deletes more exons, it helps to turn an out-of-frame deletion into an in-frame deletion, restoring the reading frame and allowing for some protein production. Nonsense or stop codon read-through therapies are designed for use in those with nonsense variants. You may remember the discussion of nonsense variants earlier. This type of genetic variant causes an early stop and terminates the growing protein. Nonsense read-through attempts to allow the body to ignore this incorrect stop signal and continue building the dystrophin protein. This type of therapy may be accessible in some countries. To learn more about variants and treatment options, reach out to your local DMD expert or genetics clinician, like a genetic counselor or a geneticist. One option for finding a genetics expert in the USA or Canada is findageneticcounselor.com, which you can use to search for in-person or telemedicine providers. The PPMD website has a lot of genetics information, and you are welcome to reach out to the PPMD genetics staff at coordinator@parentprojectmd.org for other genetics questions. We can't serve as your clinician, but we may be able to answer your questions or direct you to a clinician who can. Let us know how we can support you.